Okay. Okay, so for people in the cancer field, I think p53 needs no introduction. But maybe for a few who are not in the field, I say something about p53, just to set the background. Okay, so about 8 years ago now, Nature had an editorial about the most popular genes in the human genome, based on publications, I think popular by this standard. And as you can see, yes, can you see? Hopefully, yes. So the p53 gene, TP53, which encodes the p53 tumor suppressor, okay, right here, ranked top in this list. I believe that 8 years later, this is still the case. And the question is why? Why is p53 so popular? And probably part of the answer is that the TP53 gene that encodes p53 is the most frequently mutated gene in human cancer, and I will not argue with Matt. So Matt already said that, I can only concede. That amounts to about 50% at least of all cases of human cancer having a p53 mutation. What happens when you have a p53 mutation? Quite untypically, so I already say that top row you already know. p53 mutations in cancer are in most cases, like 70%, missense mutation, single nucleotide substitution, single amino acid substitution. That's not typical for a tumor suppressor gene, where mostly in other cases, expression is either silenced or the gene is heavily truncated. Now, several p53 mutations are particularly frequent in human cancer, and these are referred to as hotspot mutations. And one characteristic of p53 missense mutations is that in many of the cases when you have p53 missense mutations, the protein is stabilized and heavily overexpressed. This is a nodule from head and neck cancer. You can see p53 staining, nuclear p53 staining of the mutant p53, very robust, very abundant, whereas you practically see no p53 staining at all in the adjacent tissue because the amounts of p53 there are extremely low if you don't have a mutation. Now, the main outcome of these p53 mutations is the loss of wild-type p53's tumor suppressor function, but we would argue, and I think there's quite a lot of literature to support it, that at least some of the mutations, when overexpressed, may also confer cancer-promoting oncogenic gain of function. This is an area which is still under debate, but I think the evidence is quite strong that at least in particular contexts, such stabilized mutant p53 can exacerbate cancer phenotypes, especially migration, metastasis, progression-related processes. Okay. So of particular relevance to this meeting, p53 germline mutations are the major cause of the so-called Li-Fraumeni syndrome. And the Li-Fraumeni syndrome is a very, really dismal syndrome. It has a high penetrance for cancer. It's a cancer predisposition syndrome with a very high penetrance. Approximately 90% of individuals that have a p53 germline mutation will develop cancer, at least one cancer by the age of 60. Many of them will develop cancer in childhood or adolescence. So it's really a very bad germline mutation. And this knowledge is being put to some practice. And in fact, there are maintenance programs which are spearheaded by David Malkin at the Sick Kids Hospital in Toronto of monitoring children who are born with germline p53 mutations and with continuous maintenance, and this has greatly improved the survival and the overall, I would say, health span of carriers of p53 mutations. Okay. So what is the wild-type p53 doing? The wild-type p53 is a sequence-specific transcription factor. It can activate several hundreds of genes in principle, and it can also repress the expression of a comparable number of genes. Mechanisms for activation and repression are different, but I will not delve into that now. Now, this is the kind of textbook view of what p53 is doing as a tumor suppressor in response to a variety of insult signals, and you'll note that many of them are related to DNA damage, all sorts, DNA replication stress, double-strand breaks, telomere erosion, and more. p53, which is normally present in low amounts in the cell, will become stabilized, activated, and will drive a change in cell fate. And the general dogma is that in response to mild damage, p53 will enforce a transient growth arrest, allow for repair of the damage, especially if it's DNA repair, and the cells will go back into happy life. If the damage is too extreme or irreparable, then p53 will drive the cells either into cell death. Apoptosis is the classical example, but we know that now that p53 activation can drive also a variety of other programs, cell death programs, ferroptosis, pyroptosis, whatever. And or alternatively, it can drive cells into replicative senescence. Okay, now I think it's becoming more and more clear that even when present in the low levels, the wild-type p53 in unchallenged cells plays a role in maintaining homeostasis, mostly metabolic homeostasis, and energy balance, and making the cell less likely to transit into a cancer-prone state. You will notice that all of the, I mean, this is very extensively explored, covered by tens of thousands of studies, but you'll notice that all of these processes are cell autonomous inside the cell, right? And however, in recent years, I would say in the last decade, it's becoming increasingly clear that p53 functions also outside the cell in a cell non-autonomous fashion, affecting the microenvironment and also even the macroenvironment. And I would like to, okay, so loss of or mutation of TP53 alters the tumor microenvironment. And I would like to run quickly through two published examples. One of them deals with the loss of wild-type p53 in the context of breast cancer. And I'll just give you the take-home message. So loss of wild-type p53 in cancers. Before showing our data, the loss of wild-type p53 has already been reported several years ago to cause a change in the tumor immune microenvironment, and this is work by Karen Bausden, Karin De Visser, and many other groups. And this is by direct crosstalk between the cancer cells that have lost their wild-type p53 via cytokines primarily, and various components of the immune system, of the tumor immune microenvironment. We took a somewhat different approach and asked, does loss of p53 affect tumor-associated adipocytes, specifically in the context of breast cancer? Why adipocytes? Because adipocytes and pre-adipocytes are a major component of normal breast tissue. We all inject cells into the mammary fat pad, and they are also a major component of many breast cancer tumors. But because of technical reasons, adipocytes are heavily underexplored. It's much easier to work with fibroblasts than with adipocytes. So we asked, what happens to adipocytes when they are exposed to signals from breast cancer cells that have lost their wild-type p53? And this is work that was done primarily led by Ori Hassin, a very talented MD PhD student, and with the help of these two young master students. And just to illustrate the system, what we did is take pre-adipocytes, mouse 3T3L1 cells, which are a common model for adipogenic differentiation. And what you see here is staining for lipid droplets by Oren, and you can see that before differentiation, they hardly see any staining. If you drive the cells through differentiation, you get this intense staining of lipid droplets, showing that the cells have differentiated successfully. If you do the same thing in the presence of conditioned medium, from breast cancer cells that have wild-type p53, you see some attenuation of the differentiation of those cells, which is already well known. It's known that signals from tumor cells reduce the differentiation of adipocytes, of adjacent adipocytes. That's part of the characteristics of the so-called cancer-associated adipocytes. But what's remarkable, if you take the same breast cancer cells in which the endogenous p53 has been knocked out by CRISPR, Cas9, and you take conditioned medium from those p53 null breast cancer cells, it much more significantly impairs the differentiation of those pre-adipocytes to adipocytes, telling us that the loss of p53 in the breast cancer cells inhibits the differentiation of those adipocytes. Now this can be also shown at the level of gene expression, similarly. So these are two common markers of mature adipocytes. This is adipoQ, which encodes for adiponectin. This is plain 1. And you can see this non-differentiated, pre-differentiated, these genes are not expressed. You differentiate the cells, they're expressed very highly, expose them to conditioned medium from wild-type p53 breast cancer cells, you see a significant reduction. But if you expose them to conditioned medium from p53 null breast cancer cells, much more. Okay, so these are things. So the loss of p53 enhances or the inhibition of differentiation. Does it promote something else instead? So we performed RNA-seq analysis, and this looks at genes that are preferentially upregulated in the adipocytes when they are exposed to signals from p53 null breast cancer cells as compared to signals from p53 wild-type breast cancer cells. And what you can see is we have prominent activation of pathways associated with inflammation and specifically with the innate immune system. Regulation of leukocyte migration, regulation of monocyte chemotaxis, regulation of granulocyte chemotaxis, suggesting that the loss of p53 in breast cancer cells causes the adipocytes to be reprogrammed in a way that will promote the migration, recruitment of myeloid cells into the tumor microenvironment. Now, is this really the case? So we performed this kind of in vivo experiment, a little bit complicated, but I try to explain it simply. So what we take the breast cancer cells either that have wild-type p53 or a p53 knockout, and implant them in the memory fat pad of syngeneic mice, either alone or together with adipocytes that have been preconditioned in vitro by conditioned medium from p53 knockout cells, which turn on this pro-inflammatory program. So we have these 4 conditions, and what we see, so we performed cytoanalysis of the CD45 positive cells, the immune cells in the tumor microenvironment, and what we saw most prominently is an increase in the presence of cells that have markers of myeloid-derived suppressor cells, both monocytic myeloid-derived suppressor cells and granulocytic myeloid-derived suppressor cells. So these myeloid-derived MDSCs, which different people call by different names, but the bottom line is these promote an immunosuppressive microenvironment, thereby enhancing tumor growth and allowing escape from immune surveillance. And what you can see, so these are the wild-type, these are tumors that were generated by wild-type p53 breast cancer cells injected alone, and these are the ones that were injected with p53 knockout breast cancer cells alone. And you can see right away for both markers that having lost p53 will increase the recruitment of conversion of myeloid-derived suppressor cells, and thereby create a more immunosuppressive microenvironment. That perhaps is not very surprising. But if you now take those wild-type breast cancer cells and co-inject them with preconditioned adipocytes, you see that essentially it makes them comparable to what the p53 knockout breast cancer cells alone do, presumably to the mouse adipocytes, or the mouse tumor immune microenvironment. Same thing here. So the bottom line from this study is that loss of wild-type p53, at least in the context of breast cancer, will signal to adipocytes, will modify them into pro-inflammatory adipocytes, specifically causing those adipocytes as a kind of mediator to alter the tumor immune microenvironment, bringing in these tumor suppressor myeloid cells and favoring tumor evasion. This comes on top of the direct crosstalk between the cancer cells and the tumor immune microenvironment, which, as I said, has already been documented earlier by others. Okay, so that's part one. So now we go back to mutant p53 gain of function. In other words, can we see that the presence of missense p53 mutations contributes to altering the tumor immune microenvironment? And in this case, we are looking at pancreatic cancer, and this is a study that was led by Martino Maddalena, former PhD student at the lab. It's already been published quite a while ago. And again, the bottom line of the study is that having p53 missense mutations in the pancreatic cancer cells inhibits CD8 T cell infiltration into the tumor microenvironment, and presumably in this way will contribute to enhanced tumor growth and immune evasion. Okay, so here are some of the data. So first of all, okay, in this case we are comparing, right before we talked about wild-type p53 versus no p53. In this case, we are, what Martino did is he went to TCGA data and he stratified the PDAC tumors into three categories of p53 status: wild-type p53, missense p53, and truncating mutations, which we define as null, because essentially you don't get p53 protein, you get fragments. And what he looked at is he applied to this a T cell signature, transcriptional signature, to assess the abundance of CD8 T cells in the tumor. And as you can see, tumors that have missense p53 mutations have significantly less infiltrating CD8 T cells than tumors that have either wild-type p53 or no p53, suggesting that having a missense mutation is correlated with fewer CD8 T cells in the tumor. This doesn't show causality, of course, it's just a correlation. So to explore causality, we went to a mouse system using cells that were generated by David Tuvison from a mouse model of pancreatic cancer that has KRAS mutation, the signature mutation of pancreatic cancer, along with the TP53 mutation, a TP53 missense mutation. And what Martino did is he knocked down the mutant p53 in those KPC cells. So these are cells that were generated as a cell line from tumors induced in this GEM model. So the cells either have KRAS mutation plus endogenous mutant p53, or KRAS mutations with mutant p53 knocked down. And as you can see, the knockdown of mutant p53, and this is autotopic injection into the pancreas of the mice. And as you can, if you look at the staining for CD8, you can hopefully see that when you abrogate the presence or downregulate the presence of mutant p53, you get a substantial increase in the presence of CD8 T cells within the tumor, and this is quantified over multiple replicates, and there's a very substantial increase in the entry of CD8 T cells into the tumor when you get rid of the mutant p53, suggesting that here the mutant p53 has a gain of function in preventing CD8 T cell recruitment into the tumor. How is it happening? So again, we did gene analysis, and the story is more complicated. There's probably more than one mechanism, but one mechanism that was very prominent is the fact that when you have mutant p53 missense mutant expressed into these pancreatic cancer cells, this induces massive deposition of extracellular matrix within the tumor, pancreatic cancers are known for being very ECM dense, and this is believed to be a major physical obstacle for the entry of immune cells into the tumor. And what you can see is that indeed the tumors that have mutant p53, this is staining for Sirius red, which stays collagen, and you can see that they are heavily staining, very dense ECM. On the other hand, if you now eliminate or downregulate the mutant p53, you get a substantial reduction in ECM deposition, quantified significant. And we suggest that one of the mechanisms is that mutant p53, through upregulation of several genes that we identified, including several laminins, makes the ECM of the tumor much more dense, inhibiting the entry of cells, including CD8 T cells, into the tumor, and contributing to the evasion of those tumors from immune elimination. Okay, so I think this just repeats what I said. So mutant p53 induces formation of dense ECM and PDAC. Mutant p53 inhibits the infiltration of CD8 T cells, and we would argue, or we would suggest at least, that this partly at least explains this. Okay, so in the few minutes that are remaining, and I think it's really few minutes, so I'll just rush quickly to an unpublished story, which has to do with, again, with wild type p53 in cell autonomous functions. And this is a study that has just been accepted for publication and was done in very close collaboration with, was led by Yael Eilon in our lab, in close collaboration with Noah Firth and the lab of Efrat Shema. And, sorry, and again, the take-home message, if I don't have time to elaborate all the details, is that p53 regulates the expression of histone modifiers. This is wild type p53. Of course, when you lose wild type p53, this is changing. And this epigenetic modification restricts the expression of stemless genes and promotes the maintenance of differentiated luminal identity genes. If you lose p53, you activate stemness programs, and you cause a transition to a less differentiated state, less luminal identity, which in breast cancer, this is all in breast cancer, which in breast cancer is associated with more aggressive disease, less response to therapy. So just to give you a couple of highlights, since we are running out of time. So to ask whether it's already known that p53 modulates the epigenome via regulating DNA methylation and responding to alterations in DNA methylation. And this has earned p53 the title of guardian of the epigenome, in addition to its well-established title as guardian of the genome. So Yael looked at, in TCGA data, for differences in expression of epigenetic modifier, histone modifier genes between tumors that have wild-type p53 and tumors that have p53 mutations. And as you may see here, what she noted quite impressively, this is pan cancer, so it's all over, all the cancers together in TCGA, that when you have wild-type p53, you upregulate these genes, all of which encode histone H3K4 methyltransferases, and downregulate the expression of genes which encode histone H3K9 methyltransferases. Sorry, okay, I can see that. Okay, so upregulating H3K4 methyl 3, sorry, H3K4 methyltransferases downregulating H3K4 demethylases. You expect that overall you'll have an increase, when you have wild-type p53, you'll have an increase in H3K4 trimethylation. Conversely, if you look at H3K9 methylation. Genes that demethylate this modification are higher when you have wild type p53, and genes that remove this methylation are lower when you have wild type p53, and this would predict, so again, this is only correlation. Question is p53 regulating those genes in reality? And we moved to MCF9 cells as a test ground, and what this shows is that indeed, if you take either wild-type MCF7 cells or knockout MCF7 cells, these genes, and this is just a p53 activating compound, indeed, H3K4 methyltransferases are upregulated by wild-type p53. H3K4 demethylases are downregulated, and the opposite for K9. And maybe I'll just go to the next slide and then close. And does this translate into actual differences in H3K4 and K9 methylation? And the answer is yes. As you can see, in wild-type MCF7 cells, you have more K4 methyl 3 than in the knockout cells, and the opposite goes for K9 methyl 3. And since I'm over time, I just want to conclude that we looked at the genes that are regulated downstream to these modifications and performed ATAC-seq and H3K4 trimethylation cut and run. And what we saw indeed that these programs, that these modifications drive in the wild-type p53 cancer cells enhancement of expression of mature luminal identity genes and repress the expression of stemness genes. You lose wild-type p53, you flip around, cells become more stemmy, less luminal, therefore less responsive to estrogen ablation therapy, and we think that this has a number of interesting ramifications, which, okay, I'll leave for the discussion if you want. Okay, so I would conclude here. I think I'm over time already. Thank you.